think we're ready to get started. I'm Richard Scheuermann from National Library of Medicine, and I will be chairing the session on virus classification schemes. The first talk will be by Jens Kuhn, who's going to tell us a little bit about ICTV taxonomies.
Thank you. Hello again. Thank you. This is going to be a tour de force because Elliot gave me 4 points to talk about. Every one of them is a book. So I'm going to talk like super fast and try to get through this. Okay, title slide. You saw this already. Disclaimers from yesterday. I'm changing the title again. So I'm talking about classification of viruses and nomenclature of taxa. And then the overarching idea of this, and I'm not going to get into this because other people will speak later, is whether the approach that the ICTV is taking for classification could be a framework for virus subgrouping. Okay, so I'm going to talk about first topic, the ICTV process and how it all works. Thank you. What is taxonomy in the first place? Well, it's ultimately cataloging of life, then grouping them according to whatever similarities you find, ideally based on evolutionary relationships, then classifying them, which means you assign them to taxa, and taxa are established things by codes, and then you give them names, and that's the nomenclature. And on the bottom, you basically see what this means. Nowadays, you have some kind of a sequence. You have a big chaos in your GenBank or your SRA or whatever, lots of pieces, partial, not partial, all over the place. And you're trying to sort them according to something that makes sense and has some usability. And so you put all your clothes together in some kind of a cupboard. So who is this all for? So this is the first thing where probably that there's going to be disagreement here. But from my perspective, taxonomy is what it originally was. When Linnaeus started it, it was simply an awesomeness of diversity of biological life and figuring out where it all came from and where it might be going. So it's for evolutionary biologists. And so we are not a service, in my opinion, but we are our own discipline. And taxonomy is the thing that brings order into that discipline. And peripherally, this should be usable by stakeholders. And therefore, stakeholders have a very important input on especially nomenclature and how we use terms. Why is it important for the evolutionary biologists? Well, very simple things. If you discover novel sequences and you put them in your algorithm and blast again, you will find novel stuff that you haven't seen before. So you can discover lots of things. And then you can build better trees and better evolutionary trajectories, and it's just pretty. So what's the practical use of all of this? This is a thing that mainly comes out of bacteriology. There's not so much for virology yet. But remember that every new organism that is being discovered somewhere has some little secret, and it turns out they can be massively useful. So even if you're complete, outsider of the community because you're working on completely useless things like phages or something, you can discover CRISPR and get a Nobel Prize. And so the same thing, of course, will happen with the sequence space with virology. There are some examples where, we exchange Taq polymerase for PFU polymerase, and similar things can be done in virology with enzymes and other things. So who shouldn't care much about the ICTV? That's a very important point, and I want to overemphasize this. This is just like with working, if you work with a particular animal, if you work on one particular thing only, and your specialty is one particular protein and so on, then you probably don't need to care so much about the ICTV. If you have a cat at home and the species name of the cat changes tomorrow, your life will not change one bit, and you shouldn't care. Except if you suddenly decide you want to get 18 different cats and you really want to get into phylogeny. So the first thing is the ICTV classifies viruses. That brings up the wonderful question of what on earth is a virus? And this is a very, very difficult thing. And the last definition before this one came from the 1960s, which were there in Dolia. We sat down and decided we need to have some kind of an operational, with the emphasis on operational definition for virus. And that ultimately made it into the ICVV code. The ICVV code is the ICVCN. It's too long to pronounce. And the way how we derived this is by looking at all the viruses that have been classified, all the taxa that have been accepted, and then saying, okay, if we define viruses as X, what would be the impact on that established scheme? And we wanted to have the least impact with our definition. The alternative would have been to piss everybody off completely by abolishing all taxa and then reestablishing everything, and that would not have worked. And so viruses right now are basically defined as nucleic acids that can replicate and that encode that capsid protein that encapsulates the very nucleic acid that encodes it. That worked pretty much for most things. Anybody who is evolutionary, biologically interested will frown But if you do anything to this thing, we can have a discussion of what the impact will be and which study group will send rockets your way. Okay, so there's problems with this, of course. And so I'm just going to throw these out. Every single one of them is hours of discussion with the ICTV and with other stakeholders. So there's the thing of like taxa versus things, so concepts versus real stuff, which is nicely philosophical and lots of debate. There's the question of phenotype versus genotype, which is a big issue. And so what you see here is an octopus with 9 legs. And so, of course, that can't be because octopuses are defined as 8 legs. So obviously, must be an alien. Or it belongs to the octopuses nevertheless because it's a deviation. But then if your species is defined as 8 legs, it's still fine because there's 8 billion individuals with 8 and then 1 with 9. And so it doesn't change the average. Hence the category versus things thing. There's the question of, can dead things be alive? So down on the left, you see a humongous squid and it's dead and it has never been found and seen alive. So can we classify this if we don't know whether it's alive? Well, clearly it has been alive. So we should be looking into this thing. But in virology, this is very difficult when you show them only sequences because a sequence is not a virus. And if you don't have a virus, it replicates in culture. And so those are things that are still very prevalent among virologists. And then on the right, the RPs is things, evidence of things. That's when you have a partial. So you find an RDRP sequence and stuff. And well, obviously that must be just gobbledygook. Or could it be that it comes from something? And so if you look into zoology, a few bones in the desert are fine to come up with new hominid species and actually establish those. But in virus taxonomy right now, we are requesting full-length genomes. I'm actually for that. But there could be an argument made that we are missing a lot of diversity in classification schemes if we don't do that. So how does this all work from an operational point of view? So the ICTV is a relatively large organization. This is the webpage and the web address. And so you find pretty much all the information on that webpage. And this is all, and 110% kudos to Elliot, who maintains this and modifies it and listens to my and other people's complaints. You can find a lot of things in there that you would want to find. You have a taxonomy link where you can go in and you can see the newest taxonomy. You can download the history of all taxa and how they came about, all the different proposals over the years. There's a master species list. There's a iris metadata resource where you can find different strains and things that study groups find important with their different isolation designations and so on. So all of this is there. There's also the code. And so this is not from the current web page. This is only because we have pull-down menus and I could not make a screenshot of the pull-down because whenever I went away, the pull-down went away. But this is the information you find there. On the right, you see the organization. So it starts on the top with the executive committee, which are now some 20 people or so. And subcommittees. The subcommittees are listed here with the bullet points. They are historically established. Basically, we still have a plant subcommittee and we have animal negative sense and animal positive sense committees. There's overlap between them, obviously, because of the virus types. But these communities are often very different. They have different needs. And so that's why they are established. And the subcommittee chairs, they appoint study group chairs. And the study groups are established per family. And then the study group chairs have the authority to make their study groups as big as they want and invite people from the communities, or vice versa. The community members say, please, 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 I want to be in that study group, and then put them in there. And so everything there happens basically by voting on a very early stage. So in an ideal world, you have a new virus, you put a proposal in, that proposal goes to the study group, they fight left and right whether you're right with your proposal or not, and go back and forth with you and modify it and so on until everybody is happy. Then it's being submitted to the subcommittee chair. The subcommittee chair says, you are all insane. The entire process starts from the beginning. And then when the subcommittee chair is happy, it goes to the executive committee. Then again, the process starts again with the discussions and maybe modifications are being asked for. And in the end, it goes to the entire ICTV membership for the ratification. And then it becomes official. Or, it bounces back and forth for a year or 2 or sometimes 5, depending on how controversial the subject is. There are also live members and national representatives. All of them have the right to vote at a particular time. The proposals are also being posted on the webpage several months before decisions are being made so that the community can weigh in. So you can always actually see what the ICTV is up to and then loudly complain on the webpage. And then this will be taken into consideration by first the study groups, then the executive committee, and so on. 3 minutes, Christ. Okay. So there are proposals to fill out. These are very easy. These are now getting increasingly more automatized. They're not completely fill-in web forms yet, but Elliot is on the verge of getting there, more or less. And I'm going to skip all of this. I'm going to skip all of this. Higher ranks. Okay, so we have now ranks all the way up to realm. They were established based on hallmark genes. Hallmark genes, basically the idea, can we find something like a 16S rRNA in viruses? And the work here was done again with Eugene Kuhn and the people that you see on the bottom. And the idea was just like, okay, which hallmark genes, if we identify, lead to the least amount of realms, least amount of highest things. And so, for example, we identified the RDRP that basically tie all the RNA viruses together. So that was one realm. And then we find certain capsid genes and whatever. And so in the end, we ended up with 6 realms. That's where we are right now. Whether this is the ideal or correct thing is a good question. And of course, this is a scheme that will emerge. The entire process and how we got there is on this in this application. And so this is currently the entire taxonomy of viruses that you can see here. And, it's a very complicated slide. Don't look to it too much. But here you see the 6 realms. The demarcation criteria for all of this are very, very different. They're different from family to family based on history and the member composition. They're also based from taxon to taxon. So there are many different methods that are being used to establish thresholds. Different study groups like different things. There is a tendency towards certain methods that are being liked by most, and so they become ever more prevalent, and then new study groups will start to emulate. So there is a little bit of a trajectory, but we are far from having any kind of uniform approach in this. And of course, the higher you go in the ranks, the more complicated it is to get demarcation criteria set, so that we are a little bit more like the zoologists, where you say, like, if you have fur and you make milk, then you're a mammal. Okay, nomenclature of taxa. I'm going to actually skip all of this. The only thing is that's an important slide. So species names are written in Latin script, and it has a real-world application if your script is not Latin. And so here you see actually a virus name, which in this case is Ebola virus in green in Chinese. And of course, most languages have their own virus names. But the species name is always written the same way, even if you write Chinese. And so that is why we made mainly that switch. There are no rules for virus naming whatsoever. That can be a good thing. Mostly it is. Sometimes it's not. And the general thought for all of this nomenclature thing is that please, please, please do not associate meaning with names. No name is correct pretty much one day after you coin it. And so the examples here, of course, are the lady beetles on the left. I'm pretty sure one of them is not a lady. I'm not quite sure about the other. In the middle, you see an American gray squirrel. It's not gray at all. And on the right, you see a Neanderthal, but that was not found in Neanderthal. So it's not necessary to have a problem with a human virus infecting a squirrel or a squirrel virus being a human virus. So with that, I'm kind of like,
Okay, our next talk is going to be a remote presentation from Krista Queen from LSU Shreveport.
Thanks, Richard, and thank you so much to the organizers for inviting me today to talk about the method and tools that GISAID uses to identify and classify new variants that may be of interest before they become named variants. So the tools I'll talk about are part of the GISAID ecosystem, which has a seamless integration of them with our other available tools, allowing for rapid data analysis and finding variants of interest. And today, I'll mostly talk about the emerging variants tool, which is our data-driven approach to clustering and ranking mutation constellations for SARS-CoV-2 and flu. This slide is just an overview of the emerging variants tool. As I'll show in a moment directly on gisaid.org, the emerging variants tool ranks variants with looking at the unique constellations of amino acid mutations by spread and acceleration. And I'll discuss more how these rankings and calculations are made. We also have an emerging variants tool for RSV and Mpox, and we are in the process of implementing it for the viruses within EpiArbo as well. Our method for classifying variants is by amino acid mutations and their potential for phenotypic changes as curated in the different viral server tools. So for example, on the screen now, it's the COVE server tool. For SARS coronavirus 2, this includes more than 2,400 literature entries, of which around 75% are from high throughput mutational analysis studies. And so that's just a little bit of background I wanted to show, but now I'll switch over. And so hopefully you can see the GISAID home screen once you log in. We'll just go over to the emerging variants tool and let it load. So I'll start by saying the emerging variants tool monitors recently emerging SARS-CoV-2 variants that could become relevant due to signs of increased spread. We are using the existing nomenclature within the emerging variants tool, so listing the Pango lineage, but then also the constellation of mutations, which are seen if you mouse over the information. They've gotten a little bit too numerous to list directly behind the name. So we track all protein changes that were either found in at least 7 new locations in the past 30 days or found in the literature to have phenotypic effects as curated by Cove Server. All sequences that were collected in the past 100 days are categorized into constellations of protein changes based only on those 2 selection criteria. Next, we rank the constellation a couple of different ways. So first, according to the gain in number of new locations in the past 30 days. And then we also rank them according to their weighted score, which multiplies the gain in number of new locations in the past 30 days by the weighted values given to each of the amino acid changes. And those different weights are based on the type of amino acid change. And the changes can include receptor binding changes, potential drug resistance, and changes that would have structural implications. These constellations are then filtered to remove redundant constellations and then ranked accordingly. So also within the emerging variants tool, there is the ability to filter data by country of interest and also to look for specific amino acid mutations that might be of interest. And so this is the way that we rank the constellation of mutations for spread. And we'll just click over and look at how it's done a little bit differently for looking at acceleration. So to identify constellations from the above steps that are accelerating faster than the other constellations, we make use of 4 other factors, including the change of ranking in the past 7 days. And so that's the ranking that you see on the spread page and how that's changed over the past 7 days. The current ranking, the normalized gain in number of locations within the past 4 weeks, and a dissimilarity score. The normalized gain in the number of locations over the past 4 weeks is a way that we're using to factor in the rate of growth of the constellation of mutations of interest within the recent weeks. And then the dissimilarity score just measures how different the constellation is from its closest to neighboring constellations. So additionally, we have a multiple mutations tab that'll be named multi-mutes that's in the testing phase, and we expect it to go live pretty soon. This tab will highlight variants with greater than 4 new amino acid mutations as compared to all other widespread variants in the past 50 days of data. And these multi-mutes are going to be identified once they are found in 2 geographically separate locations. So this will allow us to more easily identify and monitor variants carrying a high number of mutations. So what we saw with the emergence of BA2.86, for example. The multi-mutes tab will also be helpful in the cases of new recombination events as well, since the recombination will be treated or is treated as a constellation of mutations on the previously identified variant backbone. And so these 3 tabs all show slightly different views of what emerging variants are currently circulating and could become variants of interest or concern based on the phenotypic changes. So just want to switch over to look at flu for a moment. So we have the same system implemented within EpiFlu for flu. The thresholds we use there are a little bit different. They're optimized for submission patterns. In flu, unlike SARS-CoV-2, only the amino acid changes for neuraminidase and hemaggluteinase are tracked, and they're not quite linked to individual records or within viewing the 3D structure of the protein, but that enhancement is coming soon. And you can see, if you click, you'll see all of the different amino acid changes. Then we've integrated all of the emerging variants data also with the phylogenetic trees that are available. And so you can now color the phylogenetic trees based on the emerging variant present. This just makes it easier to visualize the relationship between the emerging variants and other known variants that are circulating. And so within the tree, if you click on a sequence, for flu, you can be directly linked to the record and also to the different tools, so Audacity, Instant, and Flu Server. And so finally, I'll just say that this is our method for looking at new and emerging variants, but if there are other schemes or methods for ranking emerging variants that might work better or that you have that and you're interested in including those within GISAID, our team would be happy to meet and talk about it. And so this work is the work of the GISAID team and Sebastian Moore-Stroh's team at Singapore's A-Star Bioinformatics Institute. Thanks.
Our next presentation is by Áine O'Toole from University of Edinburgh. She's going to tell us a little bit about the development of the Pango system and software.
Thank you very much for the opportunity to speak here today. And I'm going to talk a little bit about the origins of the Pango system and the rationale for proposing it as well, and how that has kind of progressed over the last number of years, a bit about the experience of the kind of open sharing platform on GitHub, and then I'll hand over to Rachel. Our talks are fairly interlinked, and she's going to describe how the Pango system may be useful going forward. So any investigation where you have a number of virus genome sequences, so you have a question that you want to answer that may be about number of introductions, it may be about immune evasion or antigenicity, but really, in order to go around answering that question, what you want to do is build a tree, interpret it, and hopefully arise at some sensible answer, which we discussed yesterday. And so for SARS-CoV-2 from very early on, this was happening. People were getting genome sequences, building trees, and were arriving at answers. But a lot of the early studies, when they were arriving at these answers, there was lots of independent studies going on, and a lot of the studies didn't correspond particularly well to each other, and they had fairly ad hoc naming of clusters, as we saw. Sorry. Yes. So, also, another consideration is that as the number of sequences grows, and here say you've got an arbitrary deadline of when you need to have your answer by, say you need to have some public health decision that needs to be made, and you have the number of sequences growing, to calculate the exact tree, we can still only do that now up to about 12 sequences. And if we have various heuristic search methods that max out at different points, but for SARS-CoV-2, we got there pretty quickly. And by chunking up the tree into more bite-sized chunks, you can bring this time down, but only to the point that you can sensibly characterize the diversity of SARS-CoV-2 prior to chunking up your tree. So we did reach that point quite quickly. So I guess the rationale behind the Pango nomenclature was twofold. We wanted to provide a means of consistent communication across analysis, and also provide some way of alleviating that analytical bottleneck as the number of genomes continued to grow. So early in 2020, we put forward this proposed solution to this issue to provide that consistent framework for communication, that later became known as the Pango nomenclature. And a lot of the conceptual fundamentals came from Ollie and Andrew. So I'm not going to dwell on this too much because Rachel will go into it in more detail. But and also I think Betty covered it a little bit yesterday as well. But just so that everybody is on the same page, the Pango nomenclature system is a hierarchical system that reflects the evolutionary history of the virus. So here we have an example where B.1 is a sublineage of B, B.1.1 is a sublineage of B.1, and so on. Of course, these names could become infinitely long. So at a certain point, we add an alias in. And here we see C.1, which was the first alias introduced, is actually a sublineage of B.1.1. And a recent example shows that we've now reached levels of multiply nested lineage aliases, where you can, if you want to, trace back the entire ancestry of the virus all the way back to B.1 or B. But we can continue to alias this so that there's a pronounceable lineage name that we can communicate easily about. So when I was making this talk, I traced back to the kind of very first lineages that were shared on GitHub. And on the 5th of April 2020, there were 19 lineages that had been designated on a tree of about 2,500 sequences. You can see them all there. I think one thing, so we've now fast forwarded all the way to like 4 years later, and there have been more than 4,000 lineages designated actively. There are some that were removed, so there's slightly fewer than 4,000 that are currently designated. But they have been designated off of a diversity in a tree representing more than 12 million sequences. What hasn't changed between April 2020 and now is that it is still a largely manual process. It was originally annotated by hand on a tree, and still to this day, it's an individual going in, having a look at a tree and seeing what might be of interest and might need to have a label. When the original paper was put forward, one of the key factor to define a lineage was that it had to be associated with an epidemiological event. And what that event could be is really quite a flexible criteria. So in 2020, when the world was under lockdown and people weren't really going anywhere, we saw the diversity in a tree really represented geographical clustering, and primarily geography and travel histories were used to define what a lineage was in 2020. And then obviously as the world opened back up and people started to move around a lot more, the epidemiological landscape of the virus shifted. And rather than geography dominating what was a lineage, lineages began to get flagged because of the mutations that they were carrying. But because this is a flexible criterion, it shifted with the need of what we were most interested about the virus at a certain time. And this could shift again. And in applying it to other viruses as well, you would need to figure out what you were most concerned about at a given time to decide what the criterion are. So this is a figure that was created with M. Hogcroft back in 2020. Different questions about the virus will require different resolutions of groupings, like we discussed yesterday. And for SARS-CoV-2, we've had two different resolutions in play ever since the beginning. We've had the lineage system, which is, as we've described, hierarchical and fine scale, and then the clade system as well, which are usually larger and are more concerned with slightly longer periods of antigenic shift. And then, as we have discussed as well, these systems are very useful for communicating within the scientific community. But when the variants arrived and we needed to communicate with the public a lot more and with government bodies, having easy to remember pronounceable names such as alpha really helps with that as well. So the Pango system is a fine scale tracking system, and this has clearly been very useful. Having a system that's apolitical and an unlimited number of names, with not necessarily any judgment on the biology of the virus per se, is very useful. So then it's got a name, And when something of concern actually comes along, we can already discuss it unambiguously between groups. So the Pango nomenclature was put forward, but it was the structure was formalized around the time that Ollie got a grant. And now there's two official committee structures. There's the Pango committee that creates the rules of the nomenclature and reviews the rule changes. And Ollie is the chair of that. And then there's the lineage designation committee that Horizon scans, analyzes lineage requests, maintains developed software, and also updates the website. And just to point out that the Pango network has had some support. Ollie's Oxford Martin School provided funding for 2 postdocs for a year of curation, and also provided funding for the Pango network website. And then I got some money that supported the efforts of the lineage designation committee for a time, and had 2 summer students, and also managed to buy a dedicated Grinch server, and was named after the variants that stole Christmas. To emphasize that what I'm describing is lineage designation, which is a distinct entity from lineage assignment, which comes later downstream. So the assignment software, Pangolin, which originally was quite interlinked with the nomenclature system, is just one of a variety of methods that can be used to assign a lineage to a system, to a new virus. And for the Pango committee that creates the rules, luckily for them, nothing has majorly changed, so they haven't had a tremendous amount to do over the last 4 years. And Rachel will describe the new generalization of the rules in the next talk. But what I'm going to talk about is the evolution of the lineage designation system. It was mentioned and discussed yesterday, I think Richard pointed it out first, that proposing a nomenclature system is one thing, but actually updating it and having it implemented and continuing to update it is another. And having support for that system is I would say, an unsolved problem. So the sort of ecosystem around lineage designation, like I said, on the 5th of March, the first lineages were posted online. On the 7th of March, the initial commits to Pangolin, which was the first lineage assignment tool, was up on lineages.org existed. So we had a sort of series of tools that were able to provide this information to people. And the sort of flow of data in lineage designation is really quite straightforward, and looks similar enough to the workflow I showed at the beginning. We get some data, we build a tree, and we manually annotate lineages onto that tree and curate them. That information then gets pushed to GitHub, where it's open for everyone to see. And that information then goes to inform lineage assignment software. And then when people have new sequences, they can query that lineage assignment software and get useful information, and hopefully bypass the difficult steps in between. The data repository, sort of from the very beginning, has been a combination of GISAID and Colquhoun data, and combining that information was a challenge. And I think more recently, especially with the Usher tree, there's been other sources of information as well being used to inform lineages. The source of this global phylogeny has changed a lot over the few years. Originally, we had one in-house tree that we were building and maintaining, and when that became too much, we began to use Rob Lanfear's open source SARS-CoV-2 phyllo tree, which was provided for quite some time as well. Then we shifted to another in-house chunk that Rachel developed called PhyloPipe that recalculated subsets of the trees and re-estimated them as we needed them. But for some time now, we've been using the Usher tree built and maintained by Angie Hendricks. And I wanted to just emphasize, in my mind, we've had 2 really critical developments that sustain this entire process. And one of them is the availability of a global Usher tree that allows us to continuously add add new diversity into our picture of SARS-CoV-2. If I go back to that plot from earlier, what sequence placement does with Usher is completely cut out that analytical bottleneck that we were facing with SARS-CoV-2. And the other development was taxonomy by Theo Sanderson, which enabled us to view these massive trees. Before, even though we were calculating large trees in-house for, say, different major lineages, Big Tree could only view a tree of up to about 10,000 tips. So we would have to sub-chunk them to actually manually annotate the lineages onto the tree. Whereas with taxonomy now, this tree with 12 million tips can be viewed in one go. So the system, the tree has changed. The way lineages are annotated has changed somewhat. Very early lineages were using this tree. We would do a full curation and observe if there's any new lineages that cropped up, or if the definitions of previously assigned lineages needed to be updated and include more diversity. And also there was a lot of requests and emails and queries about people's sequences of interest that they thought might be new lineages. All private emails. At a certain point, we did the final full tree curation, which was in March 2021, where a team of about 10 of us curated manually 750,000 sequences. And it took about 2 weeks and caused quite a lot of stress trying to debug all of the manual curations afterwards. So that system was just never going to be sustainable, considering especially how many more sequences we've gotten since that point. So we switched to this sort of dual process where one was Chris, who was one of the funded postdocs. He would periodically go and curate subtrees that seemed to be getting quite a lot more diversity added to them, and he would add to the lineages. And then we also opened it up. So to try and harness and formalize all of the lineage requests we were getting in email, we pushed that to the GitHub and invited people who wanted to contribute or who thought they'd identified something that might be of interest and may need some sort of lineage name. We put that as GitHub issue requests. And the rationale behind that was that people who may be producing data or doing local analysis are the most invested in that, and they would have the finger to the pulse of those particular sequences. So we'd actually get there quicker and be flagged of certain lineages that needed special attention. What I didn't anticipate was that there would actually be a sort of community of people that self-assembled who are just obsessed with finding lineages. And so there's a lot of people on GitHub who have contributed, and I couldn't possibly name all of them. But by and large, they haven't been people directly connected to the data. They've been people just interested in hunting for lineages. And pick your favorite name. I think my favorite is Saltation Sentinels. But special thanks to Tom Peacock and also to Cornelius Romer, because genuinely, the last 2 years, the only new lineages that you see came from people volunteering to look at this in their spare time and flag lineages. So this is how we still have lineages like JN1. So we have GitHub also integral in how lineages are flagged. And we still have the designations and stuff that go into assigning downstream things. So this is just to sum up where we're at. 2024 now, 4 years on, we now have this adaptable system that we've kind of built through trial and error, and this has been working, and there has been lineages informing public health decisions for the last number of years. We have this amazing horizon scanning capacity. Tom Peacock, who I mentioned, flagged Omicron to the community when only 4 sequences were available. So this is genuinely, it's an incredible horizon scanning capacity. But now, and I gave this talk about 2 years ago, a similar one where I emphasized about sustainable surveillance, and we're 4 years on, and it is a system that seems to be functioning, but currently we are relying on volunteer work, which is really seems a little odd. So to go back to my flow chart from the very beginning, we have virus genome sequences, we have a question, whatever that is, and we want to try and get to an answer. And a lot of work has gone into the SARS-CoV-2 infrastructure for genomic epi over the last number of years to kind of help and facilitate us getting to that answer. So we don't want to just leave it by the wayside. It is a system that has been functioning. But we also don't want to continue to leave it vulnerable to volunteer work. We want to have a robust system. And with that, I will leave that slide with you and acknowledge there's too many people to fully acknowledge, but the Pango network, Andrew Rambo, who is mine and Rachel's PI, has obviously been integral in a lot of this as well, and Angie as well. So thank you very much.
And our final speaker for this morning is Rachel Colquhoun, also from the University of Edinburgh, and we're going beyond SARS-CoV-2.
Okay, so thank you to the organisers for such an interesting workshop. We've already touched on many of the things I'm going to cover in my talk. What do Pango lineages look like beyond the SARS-CoV-2 pandemic? Looks like there's some slight problems with the slides. Anyway, I was going to highlight the this line with quote from the original Pango paper from its inception in 2020 that says there's no universal approach to classifying virus diversity below a species. After Peter Simmons' talk and other conversations we're having at this workshop, perhaps that will no longer be true. And the work I'm presenting this morning today represents work of a number of other people, including Áine, who made some of the slides as well, and Ollie and Andrew. So the overview of my talk, I'm going to pick off where Áine left off, recapping how Pango lineages have been applied in the context of MPXV. Then I'm going to provide some more generalized definitions of the Pango nomenclature, and finally touch on some of the challenges of implementing a Pango system to other viruses. So in 2022, the world started talking about another global outbreak of Mpox, and researchers started using genomic epidemiology to work out what was going on. As we've already heard from Elliot yesterday, at the time, the global diversity of MPXV was divided into two clades, referred to as the West African clade in the bottom half of this tree, and the Central African or Congo Basin clade. However, these names were considered stigmatizing, especially as the outbreak spread globally. And so this paper by Christian Happy and many others recommended renaming the clades as Clade 1 and Clades 2A and B. Naming the clades at this level was important to highlight that they are phenotypically different and you need to handle a case from these two clades differently. But this is a virus that spills over from an animal reservoir fairly often, and the outbreak was found to come from Clade 2B. To further investigate the outbreak on a fine scale, the authors, including Áine, Cornelius, and Richard, who are here today, proposed a Pango-like system with labels to encode the genealogical relationships. And this lineage nomenclature was specific to this zoonosis event into humans and the subsequent transmission. So they called it HMPXV, and the one indicates it's the first tracked human outbreak. In this case, it was lineages A.2 and B.1 seen here in yellow, which were associated with the 2022 MPXV cases. But this again raised the question about who would be able to maintain this lineage system by designating new systems when required. And for the time being, it's being done by the next reign team, including Cornelius. And there's also a new GitHub to try and get a bit of community input where needed, if it were to take off further. Yeah. I'm going to move on now to the main point of my talk. What do Pango lineages look like more generally? Firstly, Pango lineages provide the fine-scale hierarchical partition of the phylogenetic tree. So all sequences on a tree will belong to a lineage. They're designated for the purpose of aiding genomic genomic epidemiology. There's no point creating a system if you haven't got a reason for it. We don't have any requirement for an even distribution of lineages, as there are in taxonomies, and it's not necessary to designate everything. The emphasis should be on making sure that lineages have utility. Each lineage is given a unique alphanumeric code that contains partial but not complete information about the phylogenetic history. So, for example, we've talked already about XBB.1.5, but that name in itself only tells you that you get back as far as XBB, which is a recombinant of 2 BA.2 sublineages. It doesn't tell you about the breakpoints. You haven't got the full phylogenetic history, but that is enough. We don't recommend that the lineage name should encode any other features. We think the key is in maximizing utility and simplicity. A lineage is entirely defined by the set of designated sequences, which represent that lineage. So, for example, it's defined phylogenetically rather than by a mutational profile. And here we see two trees. And in purple, you can see a lineage with the dots representing designated sequences. This gives a degree of flexibility. We touched on this yesterday in the definition, without needing to update the details of what mutations are present. For example, if a sequence is updated with a better quality consensus sequence. However, this method is dynamic, and as new information becomes available, we might want to reevaluate an existing lineage classification. So here we can see at time point 2, what we thought was a new lineage in pink really isn't that distinct. So it's okay within the lineage system to say that is no longer a lineage. These were the original criteria set for a new lineage for SARS-CoV-2 when there were just 2,500 sequences. Our requirements were that it should be genetically distinct from the ancestral lineage. There should be at least 5 high-quality genomes. There should be evidence of onward transmission seen by an internal node in the tree as it spread in the community. The lineage-defining node should be well supported, and it should represent an epidemiologically relevant event. We dropped the bootstrap value criteria when we started moving to maximum parsimony. And in practice, we sometimes did allow a little relaxation of these rules. For example, by designating a lineage before we had met these criteria. And as time went on, we allowed more epidemiological events instead of just geography. So to generalize these, we would remove the thresholds. None of the thresholds will apply in all contexts for all viruses and all sequencing responses. But a new lineage should still be genetically distinct from the ancestral lineage, with sufficient high-quality genomes to evaluate, evidence of onward transmission in the community, and represent an epi event. But more generally, these epi events could be an introduction into a new region with onward transmission, a change in phenotype, rapid growth compared to other lineages, a recombination or reassortment event, or a set of interesting mutations. To start a new lineage nomenclature given a phylogenetic tree, we recommend setting a cutoff time, and at that point, using it to separate the tree into its initial clusters, A, B, C, etc. We don't need to know that B is a descendant of A. And in fact, at the start of SARS-CoV-2, when the SARS-CoV-2 lineages were designated, it was noted that they couldn't be sure which of A and B was ancestral, but that was fine. And to save unnecessary work, this time point should not be too far back in time. It should focus on labeling current circulating variation rather than extinct variation, because that's what's going to answer your questions. To maintain human readability, we're recommending using aliases after 2 or 3 sublevels, depending on the diversity of the virus and the number of sequences you have. So for SARS-CoV-2, we had 3. For MPXV, 2 was more appropriate. Recombination is an epidemiological event. Given that there's not a single ancestral lineage, a new lineage is given the next available X prefixed lineage name. And this is a fine scale resolution. It's not going to be well suited to describing the deep diversity you have when you have host jumps. So we recommend a Pango lineage nomenclature should be at least specific to a single host. This could be a recent jump from an animal reservoir into humans. It could be applied to endemic viruses, or for a vector-borne virus, that main host is really the vector. So if it were to jump vector, at that point, you'd need to change your system. And therefore, with each new zoonosis, you create a new Pango lineage nomenclature. The next big enough MPXV spillover could be given a nomenclature HMPXV2. For reassorting viruses, the nomenclature should be based on the phylogeny for a single segment, or if there's something to justify it, use two. For example, for human flu, you could have a nomenclature for just the hemagglutinin, or also for neuraminidase. And then reassortment events of other segments would qualify as an epi event, leading to a new lineage designation. Reassortment within the representative segments would result in a next prefix lineage. Endemic viruses, there are probably some specific cases where you might want to track an endemic virus in fine scale. For example, in response to a new treatment or vaccine, or something like measles, where you've got a global spread of outbreaks. Again, we don't recommend recreating the full history of the phylogeny for endemic viruses when you start. And we touched the other day on variants. Variants, as it's been used in sort of parlance now, has a very specific signature of mutations. Lineages may help us to identify potential variants, but not all variants are going to correspond to a single lineage. Sometimes they might correspond to many lineages. For example, the first variant was the D614G mutation, which we see all across the tree, which is a really common thing for viruses. And for variant typing, you really do need to be explicit, I think, about the defining mutations, which ones you have, which ones you don't. So it's going to be a separate problem to lineage classification. So what are the main challenges of implementing a Pango lineage nomenclature to other viruses? This is primarily a nomenclature for genomic epi. It's probably most useful for teasing apart transmission chains, answering questions about whether the outbreak is being driven by community spread or seeded by imports. Towards the end, it can address whether flare-ups are related or a new outbreak. Or it could track vaccine and treatment-dependent evolution. And we can do genomic epi for viruses because they typically have short generation times, high mutation rates, and small genome sizes, which means they're relatively cheap to sequence. But the virus does need to be accumulating sufficient diversity for us to track it over short timescales. Not all DNA viruses are going to have that degree of diversity. And in particular, MPXV would not have worked if it weren't for the APOBEC editing, which occurs 20 to 30 times the rate in humans. Viruses with a lot of recombination or reassortment are probably not well suited to a Pango system. And related to the question of diversity is what sort of tree do you need? What sort of phylogenetic methods are appropriate? Bayesian, maximum likelihood, maximum parsimony. These methods can each handle different amounts of diversity, missing data. And we do really care for lineages, given how fine scale they are, about whether internal nodes are well supported. Ideally, we'd like to have that information. But parsimony may not be the best fit for a virus with lots of reversions, and IQ tree doesn't scale to millions of sequences. So you're going to have a compromise about which tree you can use. To maintain the nomenclature, we need to keep one or more phylogenetic trees. Áine already talked about this in her talk. For use for designation. For MPXV, this is being done on the next strain tree. And for SARS-CoV-2, the last couple of years have used Angie's tree. But whichever method is used, we expect there to be manual curation of the tree to check that errors are not being perpetuated due to bad data. Betty touched the other day on the sorts of things these might be, but they include amplicon dropouts, low coverage calls to reference, contamination, recombination. And what is the true tree? How well does that tree represent the true relationships between the samples? Most SARS-CoV-2 designations are defined by this team of volunteers, many of whom are amateurs, not scientists or virologists. They're doing it because they care. And there has been a paper about how to automate lineage designation based on sets of criteria. We'll hear more about that from Angie shortly. And whilst this really could reduce the lineage designation burden, manual creation will always be needed in order to make sure that the resulting lineages are not biased by the different sampling strategies. There's a huge variation across the countries in the world of the density of sequencing, and over time, the strategies changed in countries over time. And this means that the thresholds or criteria might need to be different depending where the data come from, and might need to change over time to reflect sampling density. So I don't think any Pango nomenclature can avoid a degree of manual input. It needs to be worth it with a purpose in mind. And I'd like to end with this very unscientific schematic of where we might consider different categories of viruses to sit in terms of the relative utility and workload associated with maintaining a Pango nomenclature. And I think if you're considering it, you need to ask 3 questions. Can we make it? Is the viral evolution on the scale required? Would just a tree be sufficient for a small outbreak? That might be true. What's the utility gained by using a Pango lineage nomenclature? Are we searching for new variants, global transmission chains, or immune escape? And I'm happy to talk about this slide with other people if they want to later, but because I'm out of time. So thank you to everybody for listening.
And we can open the session up for questions. And remember that Krista is online. Krista, hopefully you are still there.
I'm here.
Great.
Can you hear? This is Brian from NCBI. I think this is mostly for Áine, but at the start of a outbreak, if there was something similar to SARS-2 or something in the future, basically we maybe know some about the biology, but we don't know what's going to happen because we haven't seen an outbreak of this particular kind of virus before. I think your points about the utility of like a Pango system, I very much appreciate that, but also if you don't have the information to sort of make an informed decision about that, I'm curious if you think a Pango is a good sort of default starting place before you know otherwise, or if you would propose something else.
I'm happy to answer that. If you've got the manpower, why not? I think if you can see enough potential benefit and have the ability to implement it, then do. You can always abandon it.
And I guess partly as well, if you only have a small number of cases and you only have a small number of sequences, you probably can get all of the information you need out of one phylogeny. I guess the benefit of having a Pango system is if there's multiple groups trying to do the same analysis and you want to be able to communicate. So it does standardize that. And in an apolitical way, we're not calling things based on geography or anything like that. So I think, yeah, especially a lot of the sort of basic infrastructure and kind of trial and error that has gone on over the last 4 years, it's not that difficult to set up a Pango nomenclature. Like we did it for MPXV pretty rapidly. And standing by in case there needs to be another MPXV system set up. But a lot of that sort of back end computational stuff is already there and can be easily adapted. So yeah.
Richard.
Yeah, so I have a question for Rachel. So you sort of quite categorically said that a lineage is defined by a set of sequences. At the same time, also requiring that there is a genetic distinctiveness, right? So what is sort of the rationale for kind of seemingly excluding the possibility to say, okay, here's a parent plus these 3 mutations? Sort of.
If you've got a parent plus these 3 mutations, you've got some sequences with those 3 mutations, you can still do it phylogenetically. I think we feel like the Pango system it's trying to describe the hierarchy. It's trying to describe the tree by proxy. If you start going down mutation routes, you can have the homologous mutations all over the tree. We had variants that did that. We had variants we tracked, which were a particular lineage plus E 464K and things like that. So we had particular variants we tracked like that, but they weren't lineages because they could be in multiple places on the tree.
Question online?
Yeah, first question from anonymous participant. I did not fully understand the difference between lineage designation and the lineage and assignment. Can you clarify?
So lineage designation is the process of having a phylogeny that represents much of the global diversity and checking to see whether there are lineages. So it's the definition of lineages, finding clusters in the tree that correspond to some sort of epi event and defining it based on that. And that information is then shared and disseminated and used downstream. The assignment process is when you have a piece of software that a user uses and they have a new genome and they want to check where their sequence lies in that defined diversity. So your sequence, by, say, running pangolin, for example, isn't a designated sequence then. You've just assigned what the most likely lineage is for your new sequence. Your sequence, if you share it publicly, and it may get incorporated into the designation scheme in the future, but the assignment is just when you, say, run pangolin or run Nextclade, and you find out information about your sequence based on the designated sequences that have been predefined.
Second question comes from Mark Horseman. In previous changes, how have you managed the leverage community participant in the past? How can public health network best support tool sustainability?
So I think with the original concept of inviting from the community, the idea was that you'd have people who generated the data themselves, which possibly could be public health labs generating and sharing data, and also research labs sharing and generating data, who would have contributed issues when they spot something in their own data. As it happens, people who work in public health bodies are very busy and they've got lots of things to do and very well-defined jobs. So it's relied mostly on volunteer work of people who are very there needs to be a kind of body of people who are actually employed to look for this. And personally, I think it probably does lie in the realm of public health because it's a surveillance strategy rather than trying to rely on, academics who may get bored when the next thing comes along and then switch their interest. Or I was talking to Kuhn the other day, and he is, the last 2 years has been one of the major people who've just been defining new lineages. And he said that some people read the morning paper and other people go to GISAID and download the latest sequences and check where they fit in the Usher tree, which just seems like a very ad hoc way of maintaining a global surveillance system for SARS-CoV-2. So I think something needs to be funded, and a body of people who are employed to do this needs to exist.
So a follow-up question on it. If community member provides a feedback, is there any verification process? Is there any team which goes and verifies it before disseminating the data?
So the online community is self-regulating at the moment. There's two repositories for SARS-CoV-2 anyway. Two repositories. There's the sort of one that starts to look for things that could be lineages. And when they think there's enough evidence and they've discussed it together on those, then they move the designation over to the sort of official GitHub page with the and designate the lineages there. So there's usually discussion amongst people.
Liliana.
Hi, Liliana Brown from NIAID. I just want to bring in Krista into this conversation because it sounds some of the tools developed at GISAID were sort of automating that lineage identification. Is that correct?
Well, only in the sense of, just identifying the commonly occurring constellation of mutations that are starting to show up and helping. And if that can be used to help facilitate the Pango new designations and everything, we're happy for it to be used that way. But we're not proposing new designations ourselves.
Christian?
Hi, Christian Smolzek from JCBI and BV-BRC. I have a question regarding the Pango experts, and it's regarding the underlying tree you're using for annotations. So I noticed that the tree is actually not very well resolved. There you have a lot of internal nodes with like multifurcations, and I know it's a consequence of having not enough phylogenetic signal, but do you worry that if you're placing new sequence on that tree and they kind of fall somewhere in between, you're losing a lot of internal information, so to speak, about the relationships?
I think that's possible, but there is manual curation of the tree. You can ask Angie later after her talk about some of that manual curation. And I think that lineages are dynamic, and this means that we can, if there is evidence to show that there's something going on that's an incorrect signature, it will get picked up and it will get updated. Sometimes it doesn't matter. I think that might be an unpopular opinion. Sometimes it's close enough to what you need to get the answer you need.
No, I understand. It's usually, I mean, it might be good enough. It becomes kind of like a blast search because you're still assigning your sequence to somewhere on the tree, but just more like from an evolutionary biology point of view, you're kind of losing a lot of potentially interesting information.
But there is no other way we could do this scale of data other than with the maximum parsimony, with the maximum parsimony tree, I think. So yeah, there might be possibilities if we were to go down a different tree route. Yeah, but you couldn't do that.
Yeah, I know you can. Well, you could, for example, if you pre-cluster your sequences and then calculate using a more accurate method and then recalculating a tree, you probably could get out that kind of resolution. So maybe, I don't know. Yeah,
I think maybe we should postpone the question to Angie later because I think she's the expert.
Thank you. Thanks,
Peter.
It's very nice. I just ask a sort of conceptual question about the trees. I mean, conventionally, a tree is designed as you have designed it to show an evolutionary history of a virus, and that's actually the basis of the nomenclature. In terms of scale, though, obviously there are going to be mutations in, say, spike, which are antigenically selected for escape, and they may revert or reappear in different lineages. So how much will that distort the kind of true phylogeny that you're recording in the Pango system? I don't know what the scale is. Would one mutation make much difference?
I don't think one mutation makes much difference. Angie has manually put in reversions to try and handle things where, well, okay, maybe I should just let Angie answer the question.
Sure. Okay, with SARS-CoV-2, I think it's, knock on wood, so far it's been densely sampled enough that except for these variants that kind of come out of nowhere, we can track it mutation by mutation. And yeah, there are spike mutations that crop up in branches all over the tree. They're usually accompanied by other distinct mutations that let us, separate them onto different branches. And in fact, that helps us tell that, oh, this is really highly recurrent because it pops up with these, mutations that popped up with these and these. So we think these are all separate events. I think it makes a huge difference that with SARS-CoV-2, we had millions of sequences over time. So parsimony, I would argue, is fine when you have dense sampling like that. Now, of course, we still had variants that came out of nowhere, Alpha, Delta, Omicron. When I say out of nowhere, I mean out of the lack of sufficient sampling to catch them. So yes, we still have mysteries in the tree. We still have imperfections. And okay, Áine.
I was just going to make the point that something like this for the fine scale tracking, it's what's useful and what is functional as well. It mightn't necessarily reflect the true evolutionary history. And when we talk about homoplasies in the tree, when you have the same mutation cropping up however many times, at what point should the tree structure reflect that that is the mutation? So there is ambiguity about the evolutionary history, but in terms of the functionality of the lineages for public health, these approximations can still be useful, even if they're not entirely accurate.
Yeah, yes, I fully appreciate that. Yeah.
Cornelius.
Yeah, maybe I can say something to that as well. So in practice, we're lucky that these, even though there's 346T appearing probably 100 of times or more in the Escher tree, there are a few big clusters that kind of carry the majority. So you kind of find doing an 80-20, designating 5 lineages within JN1 with 346T, and then that does it. And there's a trade-off. I mean, we're not trying to enumerate everything on the tree, because then there would be even more lineages. There's sort of a trade-off. And in terms of in practice, it's good enough. It's enough, I think, for public health. It's enough for the people who study it with great interest. So,
last question, comment, Rodney.
Excuse me, Rodney from NCBI at NLM. So I was kind of struck by something you said, Rachel, during your talk about sort of eliminating, or not eliminating, but having a tree that only represents a single zoonotic event. And I'm curious, one, what are the practical considerations that give rise to this approach? And then two, what gets left behind in terms of informational content with such an approach? And I guess my thinking is influenced by the ongoing slow burning pandemic among animals, mostly birds and some mammals, with flu, and whether or not it would be important to have a tree that represented this entire lineage to public health professionals and others in the scientific community.
The scale of variation you're going to have when it's jumping between hosts, and I think it's not going to be well represented by the sorts of tree structure that we're likely to be using, is the main thing. It really comes down to the scale. I agree that for flu and things where it's jumping between hosts, you want to be able to capture that, but I think that probably the scale that you want is slightly bigger. What are the trends? What's the question you want to ask if you want to apply this? Have you got a bit more clear?
You're really going to be affected by sampling, because you're not sampling the reservoir.
So much unsampled diversity, and it's pretty much endless, like that unsampled diversity.
I guess I'm looking for, and I think this is a great response, it's very practical and certainly reflects the pre-pandemic state of surveillance. But I guess I look forward to a world where surveillance, both animal surveillance and human surveillance, is ongoing, and that we have more sequences. And the question I want to answer right there is the same question we just got into discussion about mutations that keep cropping back up. Understanding, I think, the mutations that are important to host shift. And then, of course, flu, we're probably talking more than just mutations. We're talking about some shuffling or some reassortment. But I think that would be, from a public health perspective, knowledge that we would be interested in, particularly if we are doing surveillance and we begin to see these mutations associated with human trophism cropping up in animals.
Go ahead.
Just to respond about that, I think that is a good point, and I think that there's no reason why that sort of information couldn't be represented by a clade system. And then I suppose the specific case of a Pango-like nomenclature could be used if, say, for instance, there was a spillover into cattle with evidence of sustained transmission that then took off, and it would be useful to have fine scale tracking when you see this evidence in the new host. So it doesn't necessarily have to be a spillover into humans. It could be a spillover into another sort of naive population, but a clade system could also be a flexible way of representing that diversity and spread alternatively.